When we're talking immunological methods, okay, everything here is going to be based on what is known as serology. Okay, ology, study of blood serum, what's in the blood. Specifically, serology is going to focus on antibodies. When your B cells, the part of your immune system is responsible for making antibodies, the B cells, make antibodies. The B cells are not making antibodies to E. coli itself. No. Your immune system does not recognize E. coli. Your immune system does not recognize Staphylococcus aureus. Your immune system does not recognize influenza virus. What your immune system recognizes is a specific pattern in the amino acids that are on the part of a protein on the surface of the influenza virus. Specific pattern of amino acids that are part of a protein on the surface of E. coli. Specific pattern of amino acids that are part of a protein on the surface of Staphylococcus aureus. Okay, see the pattern here? See what I'm getting at? So your immune system does not know the bacteria, does not know the virus. It knows part of a protein on the surface of. So because it's your immune system, your B cells, your T cells, are learning the parts of a protein, the small parts, well, it means they can be highly specific. Yes, sometimes your immune system, your B cells will make antibodies that are so highly specific that only certain strains of E. coli can be bound by those antibodies. Yeah, they can be that specific. Or, turns out the pattern of the amino acids that are part of that protein on the surface of E. coli, well, it turns out that that pattern is conserved in a lot of bacteria. Well, then that antibody is not going to be so specific. That one's going to be kind of general, binds to a lot of different bacteria. So what immunologists have done is they have studied what about proteins makes them more or less specific when it comes to a B cell being able to recognize it. They've studied it. Microbiologists and immunologists have then isolated either the B cells that make these antibodies, or they've just bled somebody, took some of their blood, and have isolated the antibodies that are highly specific for only certain strains of E. coli, that are highly specific for only certain viral strains of influenza, or nowadays with COVID-19. Why would they want to do this? Well, first, if I want to test you to see if you've ever had a certain disease, as we talked about in a previous chapter, you get sick, you get infected, there's, that dormant period where, everything's incubating in you, and then you get sick, and then hopefully, you start to recover as your immune system kicks in, and then you're recovered and the convalescent. I can test to see if you have an infection during the disease progression. Yes, I can find the virus, I can find the bacteria. But afterwards, how do I know? How do I know if you've been exposed? How do I know you've had a disease? Well, I can go and I can take a sample of your blood. Take a sample of your blood, isolate the serum proteins, get rid of the red blood cells, the white blood cells, keep the serum, the liquid portion, which has all the proteins floating around in it. Of those proteins, a lot of them are going to be antibodies. Turns out you make antibodies to everything you've ever been exposed to. So here's, it's kind of like a record of the state of your immune health. I have been exposed to this long list of things over my lifetime. How do I know? Because I make antibodies to this, to this, to this, to this, to this. If I'm not making antibodies to it, then there's a good chance I was never exposed to it. This is going to date this lecture, but if you look at the COVID-19 things nowadays, okay, there's 2 tests they can do. They can do the swab of the sinuses, find out whether or not the virus is actually there by testing using DNA tests. Now, just because the virus is there in your sinuses doesn't mean you have COVID. It just means you're right now a carrier. And we talked about carriers last chapter. You can take blood, isolate the blood, the serum, and then here I have the proteins for COVID. Here I have your serum. I'm going to use your serum and see if any of your serum proteins bind to the known COVID-19 proteins I have. If you have antibodies that bind to the COVID-19, well, that means that at some point in time you had an active infection with COVID-19. We can turn around and we can then use the serum serology in the other way. I have an unknown, don't know exactly what it is. Or I know it's E. coli, but it turns out there's lots and lots of strains of E. coli. Many of them, they're just there. They're no big deal. Some of them, though, can be killers. Bloody diarrhea, jack-in-the-box killing burger E. coli killers. So, by using serum antibodies that were isolated from patients who had that bloody diarrhea causing E. coli 0157H7 if you're curious, I can take this serum that has antibodies, and the antibodies have been tagged with either fluorescent or some way so I can visualize them in a microscope. And then I add it to the, to the plate, the glass, the prepared slide, or into the broth culture. And then I wait and see, do the antibodies bind to the E. coli there? The antibodies that were taken from somebody who had a 0157H7 infection. So now they have antibodies that are specific for part of a protein on the surface of the killer E. coli. Do they react? Yes and no. If they react, then that means that that bacteria took from a patient is the bloody diarrhea one, and they need to be treated as quickly as possible, as aggressively as possible. If they don't react, well, then that means it's not the killer bloody one, and they've got a disease that's probably not going to be as life-threatening. So we can take our time and tailor the treatment to what they have instead of, rush. So when you go and you look here in table 15.3, as we're going to see, there's many different ways to use antibodies, whether they're human from somebody who had a previous infection. We also use lab animals, rabbits, mice, horses, pigs. Did I say rats? I said rabbits, rats. We can inject them with the microbe, the bacteria, the virus, whatever. They mount an antibody response to it. We isolate the serum proteins from them, and then we further isolate the antibodies that are specific for what we want to test for.